I think what I'll do is I will post a homework assignment on photosynthesis, but I'm not going to make you do it over break. All right. It's just I'll release it. I'll give you an extended deadline, so you don't have to worry about it over break. All right. But if you wanted to get it done between now and break, fine. But I'll try to get that posted by midweek. But again, extended deadline, because you really don't need to think about that over break. And it's more for getting you prepped for the next exam in here. Okay. So look carefully at the date. So usually when I do these types of things, it'll look like a quiz, an EduCat quiz, but it's just an assignment. And it'll just be open book, open note, do it, it's like homework. But it's homework with a purpose. It's not just busy work. I want you to think about these questions because similar content will show up on the next lecture exam. Okay. There seems like there's something else I should be, some other announcement. I'm blanking at the moment. It'll come to me. All right. So photosynthesis. Let me go back. All right. So photosynthesis, we were covering that before last week, before the exam, looking at this idea of like how many photosystems, what kind of electron flow are we looking at, what are the starting electron source, what's the final electron acceptor, how does ATP get made? So no matter what organism is doing photosynthesis, these are the types of questions that you'll ask, like how are they doing it? How did they come up with this? And so really it didn't start off this way. This is normally how we talk about it in because we focus in on plants, and this is how plants do it. And then cyanobacteria also do it very similar to this. But this isn't what first evolved, all right? And there's still debate over exactly which photosystem came to be first. That, by the way, the photosystem 1, photosystem 2, they're named that just based on when they got discovered. So photosystem 1 was discovered first, and then photosystem 2, well, they realized, oh, there's a photosystem, another one, so let's call it photosystem 2. So those really don't have any evolutionary significance. So there's still some debate over which one came first in terms of when you look at how the microbes do it besides the cyanobacteria. There's plenty of microbes that just have one photosystem and not both. And so this came along later in the evolutionary process. But no matter which organism, you do see that there's some flavor of a photosystem. You have some electron flow going amongst several players that will be donors and acceptors, causing that electron transport chain to go from start to finish. Here we see that there's a ultimate electron donor. When you think about water, as when plants are doing it, they use water to split strip electrons off of that and send it into the electron transport chain. And then as that's happening, you get proton pumping at key parts. And so you get a buildup of protons, which allows for the ATP. And then on this system, you see that there's a terminal electron acceptor. In this case, it's NADP+. All right, so plants and cyanobacteria have this really nice, amazing process of stripping electrons off of water ultimately and then getting it all the way to NADP+. Because once you get that NADPH, that's great. That's a nice reducing agent. It's now carrying some electrons it can go and deliver somewhere else that's needed, and that's going to actually be the Calvin cycle. So eventually, plants and cyanobacteria will use the Calvin cycle with the help of NADPH and ATP. We'll tie that into the Calvin cycle right there in the chloroplast. The plants will be busy taking in the CO2 and making sugars. Cyanobacteria do the same thing, but remember, cyanobacteria don't have chloroplasts. They were once, what we call chloroplasts were once free-living cyanobacteria-like organisms. All right, plants and cyanobacteria doing that kind of thing. And so they're oxygenic because as they strip electrons off water, oxygen will get made. Then we started to look at some variations on that. So we said some microbes can only use photosystem 1. That's all they evolved. And so there's several different groups. I mentioned green sulfur, but there's other ones out there where they just use photosystem 1. That's all they're doing. But that's okay. When you look at how they do it, it's pretty good. They have this ability to, let me go to this one. Okay, photosystem 1, and they can use something sulfide, some external electron donor like that, where they strip electrons off of it and feed that into this short electron transport chain process. And so as they do that, they are ending up giving it to NADP plus will be at the end. So you get that great NADPH. They will make ATP off this. There is a proton gradient established, so they'll get ATP as well. So let me go back to this energetic. So this is a reduction potential diagram. And so they're showing when the photosystem 1 pigments get hit with light energy, one of those electrons off the pigment gets super excited, it goes way up into the negative portion of the reduction potential. And then you can see ferredoxin is sitting here. So how it is with respect to the reduction potential, the artist drew it precisely. And so we see that ferredoxin is up around -9 reduction potential, -0.9, I should say. And then you can see NAD is sitting down closer to like just under -0.4. So I'm going to come back to those ideas in a minute. So how they have this drawn, it's precise. They figured out the reduction potential of these players in the electron transport. So this is what we call linear in the sense that when you look at the electron flow, it's linear from hydrogen sulfide, let's say, all the way to NADP plus. So it's a linear flow. Here again, just showing hydrogen sulfide to NADP plus. Then we looked at photosystem 2, and we said like purple bacteria do this, and they are a little bit different because they don't have a linear flow. They gotta work it a little awkwardly in a way, in the sense that they don't have this, at least these purple bacteria, they don't have this external electron. They use the electron pigment, get that excited, get it boosted off to an acceptor, but then that electron ultimately makes its way back, so to speak, to photosystem 2, where light hits it again, the electron gets excited, and it just keeps zinging around. So we say it's circular. It's a circular path of electrons. And you don't see that. There's nowhere on here where it says there's NADP plus sitting at the end. Not there. Right? It's a little weird. Like, okay, why are they doing this? Well, the other payoff, think about electron transport chain. So yes, they are developing a proton gradient which allows for ATP production. But nowhere do you see any plus 3 are getting an electron, right? So the artist was accurate. This isn't happening with this type of microbe. So the problem is, look on the reduction potential. And if you see here, this Q stands for quinone. It's called the quinone pool. So it's just one of the players that's donating electrons off to an acceptor. And when you look at their reduction potential, where they fall, quinone is lower on the reduction potential than NADP plus or NAD plus. you can't just easily give an electron uphill. You're going up the reduction potential scale. You can't do that. It's against the laws. And so they can't, this final, they just keep here. Here you go, doesn't work. And so that doesn't get accomplished directly during photosynthesis if they are only using photosynthesis work. So it says here in the tan little box, it says NADH or NADH. It's made by reverse electron flow. What they mean by that is you would have to push this electron in reverse up the reduction potential scale. And by the law of how this works, always boost the electron up high and it goes down the scale. That's how we've been looking at it through all these other examples. This one, it's like, uh-oh, well, by the time final gets it, they're lower on the scale, they can't get it directly. So what they do is they actually have to expend some ATP to force the situation. So it's kind of weird. They make ATP this way, but if they want to generate NADH or NADPH through this process, they have to expend. So a lot of microbes that only have photosystem 2, a lot of times they're like, you know, it's a lot of work, expend some ATP, I'm not sure it's worth it. So often what they do is they don't bother with it. They won't get this to happen this way. Instead, they tend to be the type of microbe who will run the light reactions, get ATP. That's great. And they can do that well enough. But if they want to make NADH, they use other pathways. And then also we see these microbes tend to be the type of microbe that won't take in CO2 and use the Calvin cycle because this has to be used. That's the whole purpose of these microbes making DH and ATP, a huge chunk of why they're doing that is to run Calvin cycle. So these microbes say, you know what? I don't want to make NADH this way, it's too much work. And so that means I can't really run my Calvin cycle. So I'm going to just grab some carbohydrate from my environment and I'll just take it in and run glycolysis, the TCA. All right. And so those are the so-called photoheterotrophs I mentioned last week, where they can run the light reaction, but instead of taking CO2 into the Calvin cycle, they just eat something that's already out there, so to speak. They'll take some other carbohydrate source and bring it in. So they can do the light reactions no problem this way, but they decide, so to speak, not to run the Calvin cycle because it's too much energy input that they haven't really been able to make. Does that kind of make sense? So that idea of photosystem 2 microbes, they're the ones that tend to go, yeah, I'm not running the Calvin cycle. I don't have enough NADH to do that. I'll just take it carbohydrate and get my carbon molecules that way. And so they have, you know, flexibility, but this only having this photosystem 2 where you have that circular flow, you don't have enough energy to make that NADH. It's a little bit of a conundrum for them. Okay, so that is going back to this idea. This is another way to show it. This idea of circular flow with photosynthesis 2. And then we're trying to show again on the reduction scale, reduction potential scale, that the final is too far down. They just can't hand on plus or ATP plus. Energetically, it doesn't work that way. So they can still generate a proton gradient. They still make ATP with the light reactions, but they don't make any NADH or NADPH. So they are a little bit at a disadvantage that way. So here you're looking at some comparisons of purple bacteria, which I just talked about with that, you know, they have that quinone pool that's 2 positive on the reduction potential scale. And then if you compare them to like the green sulfur, and another group is the heliobacteria, you can see that they're further up into the negative portion of the scale when they do their version of photosynthesis. So it's very easy for paradox to give their electrons off possible problem. Right, so it just depends on what pigments they have, what photosystem they have, how they're going to work this, and if they can manage to get their reducing agents made this way or not. And then the reason that they are running the light reactions, one of the reasons is that they want to use the ATP and the NADPH to run this Calvin cycle in order to make their sugar. And so I'm not going to get into this in any detail in this class, but I mean, that's really why they're running the light reaction. They're trying to tie it into the products from the light reaction, feed it into the Calvin cycle. And the Calvin cycle is when they go ahead and take CO2, bring that in, and start making sugar. So you can see the overall reaction, the stoichiometry down there in the box. And when you see, if I say to you, C6H12O6, what sugar do you think about? Glucose, right? That's what I think. Anytime someone says that out to me, glucose. And that's totally understandable. However, I do want to make a big deal about this, that you understand that it's not glucose that they're making. It's sucrose. All right, so for sucrose, which C6H12O6 works for, explaining the basic idea that it's a sugar, but really they're going for sucrose. Then here is another version of the Calvin cycle. So I just put this in here in case you want to study a little cleaner version than the previous one. So some plants do the Calvin cycle. Cyanobacteria do the Calvin cycle. Some other microbes do this too. Then there's these other funky microbes, like the green sulfur. They don't do the Calvin cycle. They say, well, I have the TCA or the citric acid cycle already in play. I'll just reverse it. And so if you look at the TCA cycle, and it's true, if you just reverse it, you can make your sugar off of it. And so here, what they're trying to show is the reverse citric acid cycle being used to make hexyl sugars. They're saying they'll build their cell material off of that. And so it's pretty amazing. You just put that in reverse. And through that method, they can get their carbon going and get their cell material built. So kind of weird. So by the way, anytime you see fixation, CO2 fixation, what they mean by that phrase is you're taking CO2 and you're creating a sugar out of that. Okay, you're fixing it into a sugar. Yes. So, isn't that weird? But yeah. Yep. Yep. Yeah, right here. To get this to happen. You're putting it in first. So it's a little different dynamic, the thermodynamics of it. So yeah, it's kind of weird. But microbes are wacky that way. I mean, they're just amazing in their ability to adapt and evolve and mutate. And so they kind of like just take things in stride and figure it out. It's kind of amazing how quickly they can do it too, in terms of their evolution of these pathways. So all these different things we've been learning in terms of their physiology, really, when you start looking at all these different mechanisms, really what the microbes are looking for is, I need some kind of carbon source, something, because I'm a carbon-based organism, so I need to keep that going. I need some way to get energy to stay alive, and I need some kind of electron source in order to run my pathways and get all my needs met. And so that's really what they're doing. Those three big ideas. They're looking for carbon, energy, and electrons. And so you can chunk microbes, group them into these different groups. And so on this slide, I'm pairing them up. So this is the first pair. So we say that microbes can be autotrophs or heterotrophs. And so auto is self, meaning they can make their carbon themselves through taking in CO2 and building carbohydrates off them. So plants do that well. We're heterotrophs. We can't do that, unfortunately. We can't just take CO2 and make sugars off of it. So we have to get some preformed organic source into our diet. And microbes, plenty of microbes do it that way too. So that's with the carbon. Either you can take in CO2 and run your carbohydrate synthesis, or you have to get something that's a carbon-based molecule and bring that in and act on it. The other pair, the next pair, I should say, you can either be a phototroph or you can be a chemotroph in terms of your energy source. What is your energy source? Light, or is it a chemical? We are chemotrophs. We unfortunately can't just go to the beach, lay out, and get all of our sugar needs met that way. And then the next pair is with regard to your electron source. How do you get your electron? You're either a lithotroph or you're an organotroph. We're organotrophs, but there are microbes out there that can eat rocks. Lithotrophs, rock eating. And so they get electrons off of inorganic molecules. So when you're memorizing this, memorize them in pairs for what they do. One's carbon, middle two are energy, and then so those are this slide is like the big picture idea of how they're getting those three, carbon, energy, electron. Then you can start mixing these terms. So that's what this next slide is, next couple slides. So if someone says, oh, look at that microbe, it's a photolithoautotroph. So of course, science is well, big word. And so what if you start piecing that, start breaking it down, you can, photo, they're talking about light energy. Litho, well, that must be the idea that they're using inorganics for their electron. Autotroph, they must be making sugars off CO2. So if you learn that previous slide in groups like that, learn them in pairs, and then if you start putting them together, you can figure out what they're talking about. So photolithoautotrophs, cyanobacteria fit that category. Purple and green, there's lots of microbes fit this, but that's just a few examples there. Then you can say, well, there's some microbes that are chemoorganoheterotrophs. So that means they're getting their energy from chemicals, not the light. Their electrons from that as well. Heterotroph means they have to take in some organic molecule for their carbon source. So that's what we are. We're chemoorganoheterotrophs. Most pathogens that attack other organisms are chemoorgano, especially animals, humans, and other animals. Fungi are this way. Protozoa are this way. So far, all the bacteria that they've discovered and studied, they fit into these two categories. So the other two major groups are on the next slide. They are photoorganoheterotrophs. So those are the weird ones where they use light energy, but instead of running the Calvin cycle, because it's too much effort energetically, they say, nah, I'm not going to do the Calvin cycle. I'll just bring in some organic molecule around me, bring that in, and use that carbon source and electron source. And then just be aware, some can grab in CO2 if they have enough NADH or NADPH. So they have some flexibility in that regard. So purple, non-sulfur, green, non-sulfur. So you tend to find microbes doing this in polluted lakes and streams because there's lots of organic matter in those environments. And so it's very easy for them to just say no to the Calvin cycle and just get their organic carbon from nearby. Chemolithoautotrophs are the last major category. Those are, they're using chemicals, organic chemicals, and energy and electrons. Source. Autotroph means that they are able to take in CO2 and synthesize their own carbohydrates off of that. They're very often found in environments high in iron, nitrogen, and sulfur. And they are super important for doing most of the recycling on planet Earth in terms of elemental recycling. And so they're incredibly important. And they were the ones that helped build how Earth was before we came along. They were really the ones that were altering so much of our atmosphere and the environment on Earth after it settled down and started being able to hold life. Okay, so those words really, if you learn that first slide where I paired them up, if you can get that down, then you can get all this done. It's just a matter of learning how are they doing carbon, energy, and electron sources. And then the next several slides I'm not going to go over. It's just some interesting readings on what they were doing with some of the rocks coming from Mars. And so if you're interested, this isn't something I'll test you on, but I put it in here in case you were curious. I had to be curious about. By the way, microbiologists are hired by NASA. NASA like microbiologists because, in fact, way back, probably 1970s, when they were trying to figure out how do we figure out if there's life on other planets, they went to a microbiologist. Of all the scientists, that's who they went and asked, how do we do that? It was Dr. Lynn Margulis, in fact. And she gave, I have an old, do you know what VHS tapes are? Do you still know what those are? I got to get that converted somewhere. I got to get that done. But anyways, it's a VHS tape of her talking to NASA scientists where she said you don't really even have to land on the planet. You just have to take gas samples right, near the planet in the atmosphere. She says that's all you really have to do because that will tell you if there is life on the planet. Within she used how our atmosphere is telling, if someone came to Earth, they would be able to figure out there's life on this, organic life on this, based on how the microbes are doing their thing, creating this mixture of gases in the atmosphere. Anyway, it's very, very interesting talk. So they went to a microbiologist to figure this out. And when I was at that plant biology conference, NASA was there giving, science symposiums on, there was actually a fair bit of microbiology and plant biology with their symposiums. So really interesting stuff. Anyways, so that's just for your perusal. You don't have to read it, but it's interesting. I took some of the parts from the paper and kind of summarized it there. Okay. So that is kind of the major push for the physiology section of this course. I'll still be mentioning physiology as we work through some of these other topics, but it'll be not so, I guess, intense. So this is that portion of the course. And then the next topic we're due to get into is microbial growth. So that's what I'm going to start next. And it should be available in EDUCAT now. I think it's there. So I put microbial growth next because now that you've have some understanding of the physiology, you can start thinking about how they put that all together to actually, instead of, thinking about how do they build cells now, instead of how are they making their ATP and they're reducing molecules and things like that. It's like, okay, well, now when they want to reproduce themselves, how do they actually do it? So, look carefully at the Word document on what sections to read for this. I put that in EDUCAT yet? I'll have to update because this chapter gets into more than what I can cover. So I'm giving the light version compared to that. So make sure you pull the Word document up once I've updated it. Does anyone know what microbe this might be? Yeah, the E. coli, the green sheet to know it by Thursday. It's the EMB agar, eosin methylene blue. So, yeah, beautiful color. I have my Jeep painted that color. All right, so you've already learned how to grow microbes in lab, done it several ways. And so they're really interesting to study in the lab and traditionally when they first started isolating microbes back, in the 1800s, it was done this way where you would streak them out on agar. By the way, finding a substrate you could grow microbes on where they didn't eat the substrate and turn it to goo was really hard. That took a long time. It took decades to figure out how can you get this type of thing where we can observe their growth, look at their colonies, because anything you put them on, they would just eat it. They're so versatile, like grabbing nutrients from their environment. Once you use it, you break it down and turn everything to goo. So agar was the thing. The agar, most microbes can't have agar, and that's what we use this thing. And we don't synthesize it, we get it from red algae. And so there's like red algae farms out in the ocean that get harvested and then the agar gets purified from them. So agar is super expensive, by the way. That's why it's still so expensive, because we can't synthetically. And so that's one of the huge costs of this course, is just buying all the agar. So when you were streaking those plates, those microbes out on the upper plate, and then you went and incubated it, what was happening is the cells, because they had all the nutrients provided to them in that agar, they were able to go through cell division. The typical cell, when it decides I have the capability to go through cell division, they basically just double all the contents inside themselves, including making a faithful copy of their DNA and all the proteins, all the enzymes, all of that. Everything gets doubled. They grow a little bit bigger. The cell enlarges during this whole process, and then it splits in half, essentially. And we call that binary fission. The vast majority of microbes, this is what they do. There are some exceptions. There are so-called like asymmetrical microbes, soft microbes, where when they go through cell division, you'll end up with progeny cells that look different and can behave a little differently compared to the green cells there on the left are trying to show the more typical. They just grow a little bit bigger, double all the contents, split in two. And so when you're thinking about microbes growing, we talk about generations and say that, one generation is when you have, let's start with one cell, and it divides to produce two so-called progeny cells or daughter cells. Okay, so that's a generation. And then if you grow microbes in the lab and you're trying to get to know them and figure them out, you want to know how long does that take? What's their generation time? And so that's the time required for the definition is the time required for cell population to double. And so they've worked with enough microbes over the decades here that they have a pretty good feel for what the generation time is under defined conditions. So you have to state what the conditions are. Are you, shaking them on a shaking platform? Are you just putting them in broth, sitting them still? What are you doing? And so you have to have all of that detail. But usually when we work with microbes in the lab and we're trying to get to know them, we usually grow them in broth as part of this process. And so a lot of this data is given from broth cultures. And so a generation time, they often nickname this generation time as doubling time. So you might hear microbiologists say that. Anyways, we can get E. coli to double its population in 20 minutes in the lab. Okay, that's far different than animals, what they can do. All right, so doubling time or generation time required for the cell population to double, to go from like one cell to two cells, that's doubling. Okay. E. coli, if you give them all the right nutrients and conditions, including oxygen, because they're facultative, they love oxygen, 20 minutes they can double their population. This is a good general range. 1 to 3 hours is typical for the most heavily studied microbes. They say it's usually 1 to 3 hours. However, there's some outliers like Mycobacterium tuberculosis that's been heavily studied because it causes TB. that one, it can take up to 12 hours. So when we did the acid fast stain, I think we had to delay that because Mycobacterium, the one we were working with, it wasn't tuberculosis, but the one we were working with, it still was just so sluggish to grow. We had to delay when we could have you do that. And so it's heavily dependent on what kind of environment you provide to the microbe. And so you always have to keep that data really carefully. Make sure that, you get under what conditions they grow well. Others, they might grow not so well. A lot of it has to do with presence or absence of oxygen. So then if you work with them in the lab, very often we want to track this. And we want to see how they're growing and how are they doing under the conditions we provided. So it's very typical that you will grass them out. You grow them up in broths, and then you'll take a sample of that broth with the cells in it, and you'll use a spectrophotometer and take an absorbance value. And then those, you can track their growth just through looking at absorbance changes over time. So that's what you're seeing here. We have, and then the y-axis is a logarithmic scale. Okay, this is a logarithmic scale, and basically what it is doing is it's looking at population levels over time. So now you see two lines on here. One is straight. That one is called the logarithmic graph. The green one is arithmetic. All right, so you may have seen, if you've taken ecology, they talk about populations of rabbits or whatever, and you see that, they'll have this big population increase and maybe a crash, and they graph it out. Or maybe you'll see, there'll be a big population increase and then it plateaus. So you've probably seen graphs like that. Well, microbes can do a similar thing. And depending on how you graph it out, it'll look a little different. When you work with microbes in the lab, we don't like to use the arithmetic scale. We don't like to do that. It's just makes it hard. So this green line corresponds to, if you were to plot it out the y-axis as an arithmetic scale, it would give you a curve like that, curving green line. And that's fine. It's just when I'm trying to grab data off of a curved line, that's hard to do. And so you're trying to create an easier to interpret graph. And so this red line goes with the right y-axis, and it's logarithmic. So if you turn your data into what we call a semi-log plot, that's what you're looking at here with the axis on the y-axis on the right. All right, so you have logarithmic on the y-axis, arithmetic, the time still stays in the arithmetic scale system. So then we call that a semi-log plot, because it's half and half. Okay, so that's how we like to plot out data. When we're growing microbes in the lab, to track what they're doing, we use a semi-log plot. And then when you do that, you'll get part of their growth will show up as a nice straight line like that. And that's easy to pull data off. I'll show you what I mean in a minute here. So very often this is what you'll get. And this is like standard. No matter if you're working with Mycobacterium that takes 12 hours to double its population, or E. coli that takes 20 minutes, it will still look something like this. It's just the time scale may be quite different. But you'll still see this overall pattern of growth. And so you need to know it, right? I am going to be asking about this quite a bit on the next exam, because this is standard. Like I said, it really doesn't matter what microbe you grow in a broth culture. They tend to have this pattern. So what are we talking about here? Well, you can break it down. They've done different kind of blue, gray shading here into the different sections. So the first is what we call the lag phase. Okay, so this is the lag phase, where you see their growth not really doing much. So this, you see it's really quite straight, the line. So when they came in and took samples over those few hours, they kept getting the same absorbance value pretty much. which created a straight line. So you might say, well, they're not growing. And yeah, technically they're not increasing their numbers. That's true. So during the lag phase, they are not increasing their cell number. However, they're very busy. All right. So when I take E. coli out of the freezer, I have them sleeping in the freezer, and then I pop them into broth, and then we get them shaking on a shaker before you work with them. E. coli is like, what just happened? I was in the freezer. Now I'm in this broth. Okay, what do I have to work with? And we usually give them all kinds of yummy proteins, carbs. And so they're figuring all that out. And so when I take them from the deep freeze and pop them in, yeah, it's going to take them a while to figure out, okay, what do I have available? Which enzyme physiology pathways do I need to get up and running? So they're super busy assessing their environment and getting ready to take advantage of it. Okay, so it's still really active, even though the line, they're not increasing in their size, in population size. The next one is that straight line I was mentioning. We call that the log phase. We also call it the exponential phase. So number 2 here, the word exponential. So you can call it either one. You could say it's the log phase or the exponential phase. So when they are growing exponentially, and if you take any ecology, they talk about that in population. If you graph that out of semi-log plot, exponential growth gives you a straight curve, or a straight line, I'm sorry, a straight line on a semi-log plot. And that's often what we're really interested in when we grow microbes in the lab. We want to see when do they enter exponential prolongation. That's really important for a lot of studies that we do with microbes. We want to get them into that because then when you start working with those cells when they're in it, that's usually when they're the healthiest, they're the most vigorous, right? And so it's usually our favorite time. Like, okay, we got them in exponential. Now we can start running an experiment or taking them to do this or that, right? So exponential phase is a really good point of their life cycle. And so that's often why we're tracking it, waiting for them to get there. And then we can do some experiments that we're interested in. Okay, so they're very, very busy. They're taking advantage of their environment. They're doubling their population per unit time. They're cooking along, increasing exponentially. But if you grow them in a batch culture, that has a very specific meaning. If you say, oh, I grew that bacteria, that E. coli, yeah, I grew that in batch culture. What you're saying is you popped them into some broth, like in a test tube that we give you, put them on a shaker, and then just let them go. You didn't add any more nutrients. You didn't take away any of the waste products. You just let them be. That's a batch culture. And so if you grow pretty much any microbe in a batch culture, you get this pattern where after they've been exponentially growing their population, they run out of nutrients, right? I mean, eventually, if you don't add anything in, they're gonna like, okay, we used everything up. So now they're gonna shift into stationary phase, and that is shown by plateauing. Okay, so their cell numbers are no longer increasing. They're not dead, but they're just not increasing their population. So it's usually a combination of I've run out of nutrients and I'm getting poisoned by my own waste. Okay, you got both things going on. And then eventually the scales tip to the point of, okay, now I'm dying because I have no nutrients and the waste products are poisoning me. Okay, and so then they exponentially die off, which when you take absorbance measurements of that over time, it's this straight line down their population. So all the cells are starting to lyse open, break open, and that decreases your absorbance numbers. And so you get a decrease. So like I said, this is pretty consistent from microbe to microbe. You get this pattern of growth in a batch culture system. This is another way to look at it. So the green line is when they took spectrophotometer readings. Okay, so you take a sample of the broth, pop it into the spec, tell it to take an absorbance, and then you plot that over the two. The red line is kind of like another way to assess the growth. This is the viable count. That is a delayed method. Okay, what I mean by that is you got to wait for the growth data. The turbidity, when you use a spectrophotometer, that's instant feedback. You get an instant absorbance value. And so you can track it in real time, what's going on. Viable count, you've done a viable count in lab. Remember when way back when you did a spread plate where you did a small dilution into sterile water and then you use those glass spreaders to spread them out? That's what a viable count is. And so when I'm working with microbes for the first time, I do both. I do spectrophotometer growth measurements using growth curves that we just talked about, and then I also take samples to plate out and do a spread plate, and then hopefully they match up pretty nicely. After you let those plates grow and you count the colonies and figure out the population for each solution, then you can plot that out as well. Two should match pretty closely. Right? But the viable count, the downside to that is you got to wait for the colonies to grow. And very often you can't wait for what you want to do with those microbes. You want to, many times you want to get them because that's bigger than cells for your experiment. Okay, we're going to stop there and talk a little bit more about growth on Wednesday.